Education and Research, School of Biological Sciences, Bhubaneswar, Odisha. And ma'am will be giving us a talk on unveiling the dual role of eukaryotic elongation factor EEF1A2, a novel angiogenic player in breast cancer. Over to you, ma'am.
First of all, I appreciate the opportunity to discuss my work here, where researcher as well as clinicians are present. So my research, my group focuses on finding the dual regulators of different hallmarks of cancer. So idea is to come up with certain like target if when they are like, when drug is developed against them, they are less prone to resistance development. So in this direction, we have started working on this eukaryotic elongation factor 1A2. So about which the role in EMT was like established, and my group also contributed in that. And today I'm going to focus about this work which my group has discovered, that is role in angiogenesis. So what is this eukaryotic elongation factor 1A2? So canonical role is in, it's a translational elongation factor. So it delivers amino acid tRNA during translation step. But several non-canonical roles have also been reported. And interesting thing about this factor is that it has two isoforms. One is isoform 1A1 and other one is isoform 1A2. So isoform 1A1 is ubiquitously expressed, but during development in certain organs there is isoform switching, and isoform 1A2 is switched on. So these organs include neurons, skeletal muscle and cardiac muscle, and some more like highly differentiated tissue also they are being reported to have this isoform switching. But situation in cancers is different. There like multiple studies so far are there, they have reported that there is increased expression of this isoform 1A2. So just I want to clear this point that there is isoform 1A1, but in addition to isoform 1A1, isoform 1A2 is also switched on. So ovarian cancer, it was discovered first, and then in breast cancer and in prostate cancer it was found, and its role in EMT was also reported. So my group has contributed, like we have done one study in breast cancer where we found like even like if you take ERPR positive cells and triple negative cells, so it affects in both types of cell lines, but in triple negative cell line the impact on EMT is more robust. And in like brain cancers we were specifically interested because in neuronal cell 1A2 is the only isoform. So there also we found that once the expression levels of 1A2 are increased, so then there also it serves as oncogenic gene. So why we decided to look into its role in angiogenic process? So that was based upon these two studies. First one, they found like, when they treated cells with resveratrol, and there this 1A2 expression was reduced, and the CD31, which is a marker for angiogenesis, so it's an endothelial cell marker, so that was also reduced. So there was like possible connection between these, and this one gave more like clearer idea that this could be involved, where in response to hypoxia that 1A2 expression was activated. So based upon that we decided to explore its role. So as like hypoxia, the role of hypoxia, we know that it induces angiogenesis in tumors and leads to stabilization of HIF1 alpha, and that HIF1 alpha regulates like by binding on hypoxia response element, it initiates the expression of multiple genes, and these genes have many roles. One of them is important role is in angiogenesis induction. So what we did, we mimicked what happens in tumor. So we took like ERPR positive cell MCF7 and one triple negative breast cancer cell, that is MDMB231 cell line, and those cells we manipulated like perturbed expression levels of 1A2. So shortly I will be using as 1A2. Okay. So there we did like overexpression, knockdown, knockout, and triple negative because background levels are very low, so we did only overexpression. So after manipulation, we harvested the conditioned media and then we treated HUVECs. HUVECs are that human umbilical cord endothelial cells. So these primary cells we treated with this conditioned media and then we checked what was the effect on those cells. So this shows proliferation and this shows wound healing assay results. So both these they show that yes, when 1A2 expression levels were higher, they were promoting the proliferation and they were also promoting the migration, and when you knock down or knock out, it was just showing the opposite result. And we did this in vitro tubule formation assay. There also we found with overexpression there was increased tubule formation in both types of cell lines. And this platidipsin actually it is currently in clinical trial, and about this it is reported that it targets this eukaryotic elongation factor 1A2. So we use that as like proof of concept. So we inhibited using platidipsin this 1A2 levels, and that led to rescue in the phenotype. That means like when we were increasing the levels of 1A2 and we were treating with platidipsin, so almost in control versus this group, levels of tubule formation were similar. After this, we did various other assays, which included ex ovo and in vivo assays. This is the result of CAM assay. There also you can see with overexpression, lot more microvessel formation was there. With knockdown, it reduced. This is aortic sprout assay. There also we saw more sprouts when 1A2 levels were higher. And this is matrigel plug assay. So here also for both the cell lines we could see that in plugs with overexpression of 1A2 there were more redness, and with knockdown it was reduced. This shows skin wound healing assay. So with 1A2 overexpression the healing was much faster. Here also you can see that healing was much faster in this group. So after that, because VEGF is a known regulator, so we looked into that what is the effect of 1A2 on VEGF. So this could be responsible for its effect which we see in the angiogenesis. So we found with overexpression VEGF levels were increased, with knockdown or knockout the VEGF levels were decreased, and that was like observed in both the types of cell lines. And with platidipsin treatment, whatever effect we saw earlier, that was reduced. And then we looked on to what was the effect of, because HIF1 is one major molecule which regulates VEGF, and this is how it controls angiogenesis. So we found that actually 1A2 overexpression was increasing HIF1 alpha. And the other isoform we confirmed that no other isoform was not like that levels were in fact lower than 1A2, okay, if you compare with the control. Okay, so this data, the entire data shows that 1A2 was increasing VEGF and HIF1 alpha. So this is like, I don't want you to get into the details, so I'll just summarize this data. For this, like we checked what was happening in HUVECs, inside HUVECs. So when we treated with these conditioned media, within HUVECs it was leading to activation of VEGF receptor 2 and the downstream signaling, because these HUVECs also, they show kind of autocrine effect. So there was like downstream signaling, and that downstream signaling was kind of cell line specific. In MCF7 cell, it was like AKT mediated, and triple negative cell, it was ERK mediated. So we saw like again like this activation, and it was leading to again induction of VEGF, and that was giving signal to HUVEC once again. So autocrine signaling was also amplified in response to 1A2 overexpression. So when we treated with platidipsin, this effect was nullified. So after establishing that what was happening, like 1A2 was affecting HIF1 alpha and VEGF, we looked, because initial publication was like how hypoxia was affecting 1A2. So we used this hypoxia mimic cobalt chloride, and we found that with this HIF1, of course, that was supposed to increase, but 1A2 levels were also increasing. So all these three cell lines. And then we used physiological hypoxia. So there also we found that 1A2 levels were increasing. Okay, and then 1A1, it was kind of decreasing actually. And this was true for all three cell lines which we have used. And then we asked this question that it was hypoxia means reduced levels of oxygen which are affecting, or it is HIF1 which is affecting 1A2 levels. So we used one expression level for expression vector for HIF1 alpha, and there we found that it was leading to increased level of 1A2. So it was not like the lack of oxygen, rather it was HIF1 alpha which was increasing the expression level of 1A2. So to confirm our finding, we used HIF1 inhibitor, and in response to HIF1 inhibitor, we saw that whatever increase we were seeing, that was now, it was nullified. Okay, that was true for both the cell lines. So that gave us idea about that there could be a positive feedback loop between 1A2 and HIF1 alpha. So that means 1A2 was increasing HIF1 alpha and HIF1 alpha was positively increasing 1A2. So to confirm this feedback loop, we did this experiment where we manipulated three things at a time: 1A2 expression levels, hypoxia versus normoxia, and HIF1 levels. So I will again just summarize what is shown here. So whatever one thing is, there were parallel patterns between 1A2 and HIF1 alpha, and the effect of inhibitions were less prominent in hypoxic condition than in normoxic condition. So, and further, when on 1A2 knockdown background, when we put those cells in hypoxia, so the, like if you compare hypoxia and normoxia, in normoxia levels are much lower for both of them, and hypoxia these are much higher. So all this data confirmed that there was positive feedback loop between 1A2 and HIF1 alpha. So to look into the molecular mechanism that how this 1A2 is regulating HIF1 alpha, so we found actually there is this HRE element present in 1A2 promoter, and we did this electrophoretic mobility shift assay and luciferase assay, which actually showed this evidence that 1A2 binds to HIF1 alpha promoter. So to look into the other side, that how 1A2 regulates HIF1 alpha, this was one possibility, that it could be by the MYC levels. So, but the effect of 1A2 on MYC was observed only in triple negative breast cancer cell, not in MCF7 cells. So in breast cancer, this MDMB231 cell, we found that we used various chemical inhibitors for MYC, for ERK, and in response to these inhibitors, we did this tubule formation assay. So all this suggested that 1A2 activates ERK, ERK activates MYC, and MYC leads to increased levels of HIF1 alpha. So to look into the molecular mechanism in MCF7, so this was based upon previous publication, it could be affecting mTOR. So there also in MCF7 we found that mTOR was activated, and when you inhibit mTOR, it was leading to that neutralization of the effect. So this tubule formation assay also suggested the same. So this various in vivo assays, when here the tumor size was bigger, and when we used platidipsin, this reduction in the size, the difference in the size was neutralized. But you can see that platidipsin was showing a lot of significant effect on the body weight. So it has lot of cytotoxicity. And when we checked these tumors, we could see clear correlation between 1A2 levels and HIF1 alpha levels. And the inside mechanism, like whatever we showed in HUVEX, similar mechanism was observed in these tissues also. So it was leading to VEGF receptor 2 activation and c-MYC activation, because this is for ERPR positive cell derived tumors. So we checked only c-MYC and ERK, and we could see that there was activation in that. This is data from patient samples. There also we could see that here you can see that in breast cancer patient tissues where 1A2 levels are comparatively lower, CD31 positivity is lower, but where they are higher, this CD31 positivity is higher. So this work has been published in British Journal of Cancer. So if someone is interested, they can read this paper. So this summarizes the entire thing, that once like 1A2 levels are increased, so depending upon the cell type, either it could be through mTOR or ERK, HIF1 levels are affected and there is positive feedback loop, and this leads to change in VEGF level. VEGF affects endothelial cells. There it could be affecting either AKT or ERK signaling, and that affects ultimately angiogenesis. So this work was done by my PhD student Saket Patel and another student who is not in this picture, Mohammad Khurshidul Hassan. Thank you.
Thank you, madam. It was a nice and elaborate lecture, in fact.
Thank you.